Okay, welcome to the blood presumptive test for CTC 210 Forensic Laboratory. So, if you're taking this class in person, this is the lab that we're going to be doing next in our lab environment. But if you are taking this as a hybrid or an online course, there is a chance that you will not do this in our lab on the scheduled date and time. But number one, you'll either come by the lab, pick up a lab kit, and you will take this lab kit home. And I need you to keep in mind, this lab kit will have real blood splatter on articles. So I need you to handle it very carefully. Wear your PPE, which includes gloves. Use the lab doppers or the absorbent pads that we have given to you in your kit. Lay them out on your countertop, because you don't want blood contamination at your house or in your bedroom or on your kitchen counter, depending on where you decide to do the test. But if it is a hybrid course, if you don't want to take this home, feel free to actually attempt this test in our lab environment. It does not matter. We are okay with that. So if you know that we're going to be in the lab at a certain date on a certain time and you want to pop in outside of your regular student schedule because you're probably taking other classes, then feel free to do that. You can test these articles while you're in our lab environment, so that way everything stays nice and contained and you don't have to worry about taking all this stuff home in order to do some of the testing with. However, we do think it's important for you to use real articles stained with real blood, because this is kind of what's going to make it truer to life. And that is why we don't really skimp out on you as a student, because you've paid tuition to take a real class, not a fake one. All right, so with that said, I'm just going to turn this over to the lab videos. I'm going to walk you through the steps on how to do this lab experiment. There are multiple blood presumptive tests that we're going to be doing in this lab experiment. Kastle-Meyer is just one of them, and it's really one of the most common. But there are other laboratory tests that can be done for blood presumptive as well. So you're going to see a couple of different options in here. This is going to go along with our lecture videos, and we're going to end it with some hemosticks, and we're going to end it with hemotrace. So that way you can see maybe nice little convenient strips, or nice cartridges that look something like a pregnancy test that we can use in a lab environment to help determine if something is blood or not. So with that said, enjoy the show, folks. I mean, you know, I had to do it myself. I had to make sure that this is going to work. We had to assemble the kits, and I'm going to lead you through step by step each part of the test, so that way you know what to do when you get home with your lab kit. Keep in mind, the object here is on your article that we have given you. There is probably blood splatter on it somewhere. Your objective is just to find blood, period. You don't have to find all the blood spots. As long as you find one, that's all that matters. And that article is now done. So when you find one blood splatter source on that article, then you're going to keep testing that one source over and over and over using the different types of tests that we are presenting in the video here. Your objective is not to identify where all of the blood splatter is at. So as long as you get one identification or one location, that's all that matters. Go back and do the test that positively identify that spot. And it's just extra backup because, you know, we don't want to test something and for it to turn positive and then actually be negative. We're going to go back and we're going to test it multiple different number of ways. And that's what you're going to see in the lab video. All right, so with that said, I'm turning my mic off and I'm turning my mic on within the video. Woo-hoo, it's like a fourth dimension. It's a video within a video and a voice within a voice. So welcome to the blood presumptive test. If you got any questions by the time this is over, please feel free to reach out. Otherwise, we will have your kit ready, and you are more than welcome to pick it up and take it with you whenever you're on campus. Unless you want to do it in our house, and that's perfectly fine. We'll have our lab doors open. All right, guys, you are getting ready to do the lab experiment on blood presumptive test. So we're going to go through some maybe articles that have submitted to the laboratory to see if they in fact have blood splatter on them, and we're going to have to identify if that blood splatter is there and maybe a location of where we found it. So you're probably going to get a packet that looks like this in a nice tidy little Ziploc bag. And this little take-home kit or in-house lab kit will have a few things. First, I do want to say, please wear your gloves when you do this lab experiment. And the reason is because these articles are splattered with real human blood. And I'll repeat that again. It is real sample that you are working with. And I don't know where the blood comes from. Right, I have no clue who this blood donor was. The only thing that I know is that a blood sample was submitted to us. We used it the way we needed to use it, and I have no idea if that blood was actually clean, if that's a word or not. So please wear your gloves, and please, there is a lab dopper. They're absorbent pads, but we like to call them lab doppers. So these lab doppers that are on the inside of this bag, make sure that you take that out first, and make sure that you spread it open and cover your workspace, because we don't want that blood contamination to get on the countertop. So if you're doing this at home, that is something that I need you to do, because you don't want to contaminate your tabletops or your countertops when you do this experiment. And if you're doing it in-house in the lab, then we also don't want you to contaminate our lab countertops either. So lab doppers are going to be very important. On the inside of the Ziploc, you're going to find brown bags, and the brown bag is an evidence bag. And you probably can't see it in this video, but there's some writing on the evidence bag, and it's going to change depending on the year that you do this. So just make sure that you go through and you maybe write down the information that you need for your data sheet. This evidence bag is going to document the article or object that's on the inside. It's also going to give you details on who obtained it first and put it into the bag. And then there's also a chain of custody, or what they're calling on the bag a chain of possession. So this chain of custody or chain of possession is going to document who handled this particular article from the moment of collection all the way through the moment of testing. So you might need to know some of that information as well. And again, that's going to be stamped on the brown bag when you get it. So those details are very important, and you'll want that for your laboratory data at some point in time. So this is going to be your lab kit. There's going to be articles in here that we're going to have to do some testing on. We're going to see if we can find any blood splatter on them, and if so, we're going to circle those areas where the blood splatter is located, and we'll do a few tests on that particular type of spot once we find one, and I'll give you some tips and tricks on what to do in the upcoming videos. So I'm going to use this article that has been submitted to the laboratory as an example. Yours might not be that easy, and the reason I like this is because it is white, and blood splatter or some types of dyes or indicators or stains or whatever the case is can easily be found on a white article of clothing. So as you know me, I'm probably going to stray away from giving you a white article of clothing, and if so, you're probably going to have blues or blacks or reds maybe, and make me a little more difficult for you, which is all in the name of fun, really, not torture. So when you get the article of clothing or the object that has been submitted to the lab, it's going to be in its own biohazard bag. These are biohazard bags because there is blood probably on this, and we don't want to take any chances. So we seal it up, we identify it, so that way you know, yeah, you got to pay attention to it when you use it, and you got to make sure that you know, everything is legit as far as PPE goes. The bags, mine was already broken. I kind of skipped ahead on you a little bit. But the bag will have a sticker on it, and that sticker, if I can zoom in really quickly, will be a sealed evidence sticker or something similar to this, and it says do not tamper. So that means that this article has been obtained at the crime scene maybe, and this article has been submitted through different various hands throughout the time of collection and transferring, and this sticker has to be intact when you get it. And the reason is because we don't want anyone to taint the object or the article of clothing that we're trying to test. And that seal is very, very important. So when you get it, that sticker's probably going to be there, and I need you to kind of take your finger or take a sharp object. It doesn't take a lot of effort to rip that tag, but you're going to rip that tag open, and that allows you to open up the bag now, so that way we can go through and do some testing at this point. All right, so when I go inside of this bag, what I like to do is I'll open the bag up like this, and the problem or the issue here is that I don't really want to cram my hands down into the bag, because if I do that, then I have a chance of contaminating my arm or things that are not protected by the glove with a possible blood sample. And that's, going to be a no-no for me. So what I like to do is roll the bag instead, and I will roll the bag down comfortably enough where the article is like right there up front and center, and I can just grab a hold of it and pull it out. All right, and then I'm going to sit this bag over to the side. I try to keep it on the lab diaper as much as possible, because again, that could have some blood contamination on the inside of the bag, and I don't want that to come in contact with any surfaces at my house or in the lab environment. So what I've got here is like a white article. This is actually a pillowcase that was submitted to the lab, and I like this because the pillowcase is going to show up a little differently, and I want to talk about what some of this actually is going to be. All right, so as you can see on the pillowcase, there are clearly red stains in some form or fashion on the pillowcase. So I see a large stain here. I see some stains up here in the right corner. I also see a few like little splatters that are going to be here. And then if I flip this over, I'm going to look on the other side, and Jesus, there's more. Look at all of this. So of course the yellow stains here that you're seeing show up on the screen, those yellow stains are not blood. That of course is pretty much common sense. It's going to be a reddish or red-brown color. These that you're seeing here on the pillowcase, they're very bright red. I guarantee you that those are not blood either. Actual true blood, when it dries and if it's a little older, will not be bright red. So actually I'm looking at this, and this could be anything. It could be a wine stain, it could be Kool-Aid, it could be anything that's red. It doesn't really matter, but my eyes are not really going here first, and I need to keep that in mind when I begin to test the articles that you do, right? So if something is bright red, or if it is coming off bright red on your Q-tip when you begin the test, that's probably not real blood, and you don't need to go any further. However, up here in the right-hand corner, I actually see two spots that are located right here, and they're reddish, but they're brownish. And that is a very good sign that that might be a possible blood splatter. So I'm looking for a red, brown, or totally rusty brown color. That probably is going to be a blood sample for me, and maybe not the broader reds that you see here on this pillowcase. Now, once again, your articles will not be white most of the time. They'll be something else. So that is why I like to do that, because this is very easy to kind of pinpoint where something's at. And if you've got a colored article of clothing, then that's going to be a little bit more difficult for you. But you can still see differences in them. If you look really close, you can see some intensities of the colors. And if something looks very vibrant and red to you on that garment, it's probably not what you think it is, blood. It's not that. So in the next video, we're going to focus on these areas, and I'm going to Q-tip this bright red spot to show you what that looks like on a Q-tip, and then I'm also going to Q-tip these brownish red colors that you see here in the corner. Alright guys, so in our very first blood identification presumptive test, and presumptive again means, yeah, it probably is but we can't guarantee it. There's no like, I'm going to put all of my money on the gamble. No, I'm not going to do that, but I'm going to put a lot of my money on the gamble because there is a very strong chance that it is. But there are false positives that can happen. So with the Kastle-Meyer test, if I read the directions that you're going to get in your lab kit, it's going to say a few things, and I'm just going to go through these. I'm not going to show you the page as I do them, but I'm going to go through them as we do this very first test together. And the Kastle-Meyer says find a suspected stain. So we're going to test this bright red, and we're also going to test this kind of dingy brownish color up here in the corner. And it says add DI water to the end of a long-stemmed Q-tip. It doesn't have to be long-stemmed, but we're going to give you Q-tips in your lab packets when you pick those up and you get ready to test these articles. And a long stem Q-tip will look something like this. Regular Q-tips would work, there's no problem with that, but long tips allow us to get away from the surface a little bit better so we don't, again, contaminate me and contaminate the article. And it's going to say wet with a little bit of water. So I'm going to use distilled water. That's actually what it says here, add DI water to the end of a long tipped funnel. But there is a, we're not going to give you that kind of a big bottle to take home with you. Could you imagine clunk, 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 all of the pounds that you got to do when you walk it to your car? So instead, we will give you dropper bottles of these reagents that you'll need, and here's a dropper bottle and it says DI water on the front. So what we can do now is safely transport these. I don't have to worry too much about it, and I'm going to suck up a little bit of water in the dropper, and I'm going to add a little bit of water on the end of the Q-tip. All right, so we want this surface to be wet when we do the presumptive test, no matter what presumptive test it is most of the time. And let's say that you did not know any better, and let's say that you were going to swab this area of the article or that piece of clothing. So with the wet Q-tip, it's going to say rub the suspected stain. So I'm going to rub this and I'll pick this up. Look at what this looks like, right? So I'm going to zoom in just a little bit, clear that up, maybe get it into focus if I can. And this Q-tip has immediately went red. That is a sign that that is not blood, that's something else. Blood normally does not do that, at least that dark at least. Now, maybe fresh blood would do it, but if we're talking about blood that has made its way onto a garment that's been there for a little bit, it's dried, it's crusty, it's made its way into the fabric, blood normally does not do that at all. So I'm not actually even going to test that area, because that blood color or that red color is a sign that that is false. That is not blood at all. Now, of course, if you can imagine, as I rub the yellow spots, nothing's going to happen there either. So now I'm going to go up here to this more of a brownie color that I told you to pay attention to, and I'm going to take my dropper bottle yet again, and this is what you'll need to do at home or when you do this in lab in person. So we're going to add a few drops of water around the end of the cuvette, or a cuvette. Woo! I just got out of UV lecture. It's not a cuvette, it's a Q-tip. All right, and then I'm going to take this Q-tip and I'm going to rub it, and you do not have to rub it gently. I mean, you can really like get in there and rub, 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 rub. I mean, that's what you need to do right there. All right, so when I do that, look at my Q-tip here. I don't know, again, if you can see that or not, depending on the light. But that Q-tip really doesn't have anything on it color-wise at all. If you do see something, it will be like dirty looking or dingy. That's kind of what it would look like. But other than that, I really don't see anything on that at all. All right, and that's good, because that is a sign, again, that that really is blood that we have on the end of that Q-tip, possibly blood. All right, so the next step of the Kastle-Meyer says once I do that, I'm going to add 1 or 2 drops of ethanol to the end of this Q-tip. So here's ethanol. It will also be in a dropper bottle for you. And I'm going to add a few drops of ethanol onto the end of the Q-tip. It doesn't take rocket science. Just don't come in contact with the Q-tip, please. and I like to go all the way around the Q-tip. So 2 or 3 drops of ethanol is all that's going to be needed here. So again, no color change has happened. It's not supposed to at this point. And in the next step, I need to add a few drops of Kastle-Meyer reagent. So I'll do that in the next video when you come back. Alright, so in the next step it says add your Kastle-Meyer solution. You're actually going to not find a big jar like this. This is what I have. The Kastle-Meyer is going to be in a small dropper bottle like this one as well. The Kastle-Meyer reagent needs to be clear. It should not have a pink color at all. Think phenolphthalein pink, because that's kind of actually what's in here. It should be a very clear solution. If your solution is not clear, then that means the Kastle-Meyer reagent is bad, and you should not be using that at all. So I'm going to add, the directions say, a couple of drops of Kastle-Meyer reagent to the end of this test, or to the end of this Q-tip. Alright, so when I do that, here's 1, 2, and again I turn the Q-tip around, get it all nice and moist on all sides, because to tell you the truth, I forget which side I rub on the garment. So I just turn and I apply the reagent to all of it. Alright, so notice the Kastle-Meyer reagent's not doing anything on the end of the Q-tip either. It's not supposed to. So if it does do something here, something's off, something's not appropriate, Kastle-Meyer's just not working the proper way. Color changes should not be happening as of right now. Alright, and then the final step of the Kastle-Meyer test is to then take 1 or 2 drops of hydrogen peroxide. Again, you're going to have hydrogen peroxide in a dropper bottle, and I've got it just here differently, and I'm going to add a few drops of hydrogen peroxide to the end of my Q-tip. And as you see, automatically, look at this, I'm getting a color change that begins to happen here with the Q-tip that I have in my hand. So I'm going to try to zoom in if I can, and that pink color that you're seeing develop on the end of that Q-tip, it was colorless up until this point, but that pinkish color that you're seeing now is the positive test for blood based on the Kastle-Meyer. So what's happened is that now I have identified an area on the garment that is blood possibly. And from this moment, I'm going to work with that same spot, and I'm just going to go through the different types of tests that we can do in the laboratory environment other than Kastle-Meyer. This is a great test. I mean, it works great. But by no means is it the only test that we can do for blood identification. So absolutely, we're going to say that area right there is probably positive for blood, and I'm going to swab that area again for the rest of the test, because that will be enough blood there in order to get me positives, if in fact it is truly an actual blood sample from a person. So I'm going to list positive, I'm going to write down the data, I'm going to write down my observations, and then I'll move on to the next test that we've got to do with the blood identification. So Kastle-Meyer is easy breezy, and now it's done. Alright guys, we're back at it, and we are on to the next part, and this is called leukomalachite green, or LMG test. It is another presumptive test that can be done in a lab environment. It is particular to blood. It can detect other things, but blood is one of those common things that we'll see. What you'll find on your dropper bottle, again, I don't have a dropper bottle, but you will, it will probably say LMG solution on it. LMG solution, if you look at this, it should be colorless. It should look kind of like water. You might see a little tinge of like a greenish or a bluish in here, but it will be very faint. But overall, there is no color that's associated with that solution, and there should not be. If there is, then we should not be using that reagent. It has spoiled, and we need to make that fresh. All right, so that's why when we tell you to pick up these lab kits, you need to do the lab quickly, probably on that same day, so that way these reagents are not going to spoil on on you. So with the leucomalachite green, or LMG, again, it's going to say wet the end of a Q-tip with a little bit of water or ethanol. Actually, either one of those will work. I'm going to use water. Why not? I mean, water we used in the last one. Water actually works for these. So if you want to use ethanol, you can. It doesn't really matter. And we already said that was probably positive. So I'm going to rub the end of this Q-tip and I'm going to push down. I don't lightly tap it, like really get in there and kind of rub. You want some of that sample to be transferred onto the end of that Q-tip as much as possible. And then once I add the water, it says add 1 to 2 drops of LMG solution. All right, so I'm going to kind of come up here and see if I can focus in a little bit now. And with our LMG solution, I'm going to just simply add it using a transfer pipette. But once more, you'll have water or in a dropper bottle, just like your water and just like your ethanol and your peroxide, but this is LMG. All right, so if I can get it into focus right there, we're going to add a couple of drops of LMG, and notice nothing really is happening right now, but I'm turning the Q-tip so that way I wet all surfaces. Once more, I don't know which side I swabbed, so I'm just going to make sure that I get all of them that I can. And then the next step, it says wait for 10 to 20 seconds. No color should develop. So as I'm sitting here and as I'm waiting, I'm not really seeing any color, but I'm going to go get another transfer pipette because I need to add peroxide in our last step. And then we're going to take a look at the end of our Q-tip when I come back. So just give me 20 seconds and I'll be right back. All right, so that was shorter than 20 seconds, but that's fine. We probably have had 20 seconds to go by since we added our LMG solution. All right, so then now the last step says, if no color change is seen and I don't see any, then we're going to add a few drops of peroxide on the end of this Q-tip. And as I do that, I then kind of wait here for a minute and look at this, folks. That teal blue color that you are seeing here on the end of that Q-tip, that is another positive identification that yes, in fact, we did get blood. So just like the Kastle-Meyer, it is a presumptive test. It is a different type of presumptive test that we can do in the lab, and this is very evident. And the reason that people like this one is because look at this color change. It is like a blue teal green. I mean, that's what we're seeing here, and it's very dark. Blood does not look like this, right? And Kastle-Meyer has phenolphthalein, and phenolphthalein turns pink. And some people get weirded out because they're like, oh no, is that pink from the Kastle-Meyer? Is that pink from the blood? I don't know. Well, LMG solves that problem because as you can see, that is a totally different color than what you would associate with as blood. All right, so that is positive for blood, presumptive positive. Meaning, again, there's no guarantee, but there's a really good possibility that that is, in fact, a blood sample on the garment. So that is the leucomalachite green, or LMG test, just as easy as Kastle-Meyer, and we're going to continue on with a different test in the next video. All right, so we're back to the same article, and that's my pillowcase here, and I need to take a new Q-tip because we're getting ready to do a new test, and that new test is called tetramethylbenzidine. That is what this is, benzidine. So if I try to get that into focus if I can, that is a TMB test. And the TMB test is just as easy as all the others. It is wet your Q-tip, add the reagent, and see a color change. So once more, it's going to say find the suspected stain, and we have, and then it's going to say add a little bit of water onto the end of a Q-tip, and that's what we're doing here. So now I've wetted the Q-tip so that way it can come in contact with the blood. And then it says to, look at me, I broke the Q-tip. It's going to do that number onto the blood stain. We want to make sure that we rub kind of hard. Again, you want some of that sample to be transferred onto the end. And That end of the Q-tip, again, it is a little dingy, I guess, if you want to say that. So I'm going to zoom in here and try to focus in on the Q-tip area. So there's a little bit of color, not much, just a tinge of brown there on the end. And that's a good sign that, again, it is truly a blood sample, possibly a blood sample. Next, it says add 1 drop of TMB reagent to the end of that test tube. TMB is actually going to be one of the few that you're going to find that actually has a color to it. So it's like Slimer green. If you've ever watched the Ghostbusters, this is what I think Slimer would look like. So we're going to take some of Slimer's juice, and that's TMB here, and it says add 1 drop of TMB reagent on the Q-tip. So I'm going to add, as I turn, 1 drop on each side just to get that reagent in there, so that way it can start reacting with the possible blood sample that we've got. And then our next step says add 1 drop of peroxide. Now TMB has an odor, so I want you to be prepared for that. It is very strong, vinegary smelling reagent, so don't freak out when you add a few drops of that on your test tube. It's going to happen. And then we're going to add a few drops of hydrogen peroxide on the end of this Q-tip here, and ta-da! Look at that. Look how pretty that color is. It is almost just like the previous one that we did, but tetramethylbenzidine or benzidine, TMB, is also going to have a drastic color change. So again, this is a presumptive test, and that possibly means that that spot is really blood. And now we've got 3 presumptive tests that are telling us the same story. So that is really good. They're agreeing and they are getting along with each other. I don't have any tests so far that has turned out negative, which means I'm on the right path. So if blood is present on this Q-tip, that's the color that you're going to see. It is a very blue-green, deep color within 20 seconds. And for us, it happened almost automatically. So we're finished now with the TMB test, and now I can move on to the next step. So we did Kastle-Meyer, and we did leucomalachite green, and now we did tetramethylbenzidine, and now the next one is our hemastick and our hematrace. All right, guys, so what you're seeing here is a original container that has hemasticks in it. Now, these are a little bit more convenient, but they're still presumptive here. So that's the thing that most people are confused with. Hemasticks look all prim and proper, and it's like, yeah, that's blood, but you know, it's still presumptive. And the reason it's still presumptive is it still works by an indicator. So I'm going to open up this container, and on the inside you're going to see some strips, and those strips really look like pH strips, honestly. I mean, that's kind of what they remind me of in a way. So I'm going to take one of these strips, and it looks like this. And on the end there is like a yellow type of pad, and that pad is an absorbent pad. Let me zoom in for you. That pad is an absorbent pad, and that absorbent pad is what needs to come in contact with your sample. So to use the stick, I'm going to take a Q-tip and I'm going to wet it with DI water, and then I'm going to rub it onto the stain that we keep going back to, and I'm going to touch the end of this stick with the Q-tip, and we're going to see what happens. So the reason it's presumptive is because there's indicator already built in to the pad. So this is no different than some of the other presumptive tests that we do with indicator and color reagents. There's no difference here, which is why this is still presumptive. So in the next video, I'm going to zoom in a little bit better. I'm going to lay this flat so it doesn't get bouncy and make you dizzy and make you drunk. And then we're going to see what happens when I wet that little spot and apply it onto the surface of the hemastick. Alright guys, so for the HEMA sticks, it says, yet again, go figure, take your Q-tip and then wet the end of the Q-tip with DI water or with ethanol. So I'm actually doing that on the side because you've already seen me do that process 3 different times already. And I'm going to use DI water again. But if you want to use ethanol, I don't care. And then I'm going to go back to that same spot that we have dabbed and swabbed in our other 3 tests. I'm going back to the same, doesn't really matter, and I have now rubbed my Q-tip onto that stain. I don't think that you need to see that, but I did it. Trust me, I did. And then it says take your Q-tip, and then we are going to touch the end of this pad with the Q-tip. And in doing that, look at what's happened. I have now taken my blood and I have touched it onto the end of that pad, and that pad is now turning color. And folks, it is very similar to the color that you are seeing in our other reagent tests. So our LMG test and our TMB test, some of those were like deep blue, but you know, there's some green that's going to be there as well when you do those tests in person. And that's what's happening here. It starts off green, and then sooner or later it is going to go more of a blue tinge on you. So this is a very easy, simple, convenient presumptive test. No reagents are needed with the HEMA sticks, and that's why we lock them. And to be honest with you, the HEMA sticks very often is used for urinalysis. So if you're peeing in a cup and they want to know, is there blood in your urine, they can use these sticks and dip it down in there or apply the urine to the stick to see if it turns color just like this. And if they get that bluish, deep blue or green color, teal color, then that is positive. Yeah, you've got blood in your urine. But guess what? It is positive for articles of clothing as well. So forensics labs can sometimes use HEMA sticks just like this. And again, it is one more piece of evidence that we can add to the pile of evidence that is getting collected, meaning, yeah, sure enough, that probably is a blood sample for us. All right, folks, so one of our last blood identification tests will be the HemaTrace test, not HEMA sticks. HemaTrace is a little different, and the reason is because HemaTrace is often regarded as not a presumptive test. This actually is very specific to blood, but the problem is that it's more on the expensive side of the house. So if we can take care of trying to identify where things are with cheaper, easier obtainable reagents, and that's what we're going to do. And then if we really do want positive confirmation, HemaTrace is where it's going to be at. So HemaTrace, this is actually just a box, a kit that we order, and on the inside of the HemaTrace box, there's going to be an assortment of bottles that have buffer on the inside. That buffer is self-proprietary. They do not like for you to know what's in the buffer. And then here we have a sealed cartridge that we're going to open up in just a minute with one of our tests that we're going to do, so I can show you how this is going to work. But there are typically these kits, there are 25 to 50 cards that are inside of the box. We order them in 25s because we just don't use them a lot, and that typically will hold us over for 1 or 2 years, and then when things expire, we order new ones. All right. So this is our beginnings of the HemaTrace. So what you will see in your lab kit when you pick that up is a buffer bottle that will look very similar to that. It will have some type of squeezy bottle. And then a packet that will look like this on the inside, and that packet will have your card on it. So what I like to do is get things ready first. I like to wait until I get ready to do this test, and then I'm going to open up this pack, and on the inside of this pack is just a tiny cartridge. That's all that is right there. There is some identifiers onto the cartridge, and again, I don't know if that can actually show up on the video or not. But down here at this spot, or this circle, that is where you're going to be applying your sample eventually. And then up here in the top portion of the cartridge, you will see a T, and the T means test, and then you will see a C, and that C means control. So you will see a positive test every single time at the C location, because that means that this cartridge is working the proper way. So we will see a line that begins to develop at the C, and if I see that line, then I look at the T line, and is it there or is it not? If I do not see a line here at the T mark, then that means we have a negative test. If I see another line that develops at the T, then those two lines mean a positive test. So think of this as like a pregnancy test in a way. So two lines are what you want to see here. Maybe if you're doing a pregnancy test, you don't want to see two lines. But when we do forensic blood identification tests, we want to see two lines on this cartridge. All right, so the way that this is going to work is that the directions are going to say, get a swab. Oh, well, go figure. And it says, obtain the buffer. Well, go figure. Right, there's my buffer. And then it wants you to actually not wet your Q-tip with water. It wants you to dip your Q-tip or to wet your Q-tip with the buffer on the inside. So I'm going to take my Q-tip, and I'm actually just going to quickly insert it into the buffer, because I'm not actually in the level yet. I'm just kind of getting it ready. But I'm going to quickly insert it in and pull it right back out. That's what I'm getting ready to do. So I just need it wet, just damp on the outside with the buffer, and then I'm going to put this top back on so I do not accidentally spill that container. I'll need it later. And then you're not going to see me do it on the screen, but I'm going to, guess what, rub that spot that I think is blood on my pillowcase that they gave me in the lab. So here I am, rubbing all over again, and I get a little bit of tinge of color on the end, and that's okay, it's not a big deal. And then the direction is going to say, all right, now take this buffer tube again, and we are going to open it back up, so that way I did not accidentally spill it over the course of time. And then it says, insert your Q-tip in the inside and let it sit. And then it says, jiggle it around. So what you want to do is you want to have that Q-tip come in contact with all of that buffer as much as possible, and you're kind of wanting to slosh off some of that sample that you've just applied on the end of that Q-tip. You want it to slosh off and go into the buffer. So it says keep it vertical, and then we're just going to kind of stir for 10 seconds. And then it says make sure that you press the Q-tip up against the sides of the container on the inside. So I'm pressing it up on the upper regions against the side, so that way we're squeezing out any remnant of stuff that's left over in that Q-tip. And then I'm going to put the cap back on, and then my sample is ready to go. So that's the sample that I'm actually going to apply in the next step. So from this moment, that is my sample, not the Q-tip, all right? So at this time, I'm going to take this lid up here at the very top. Again, I don't know if I can focus, but I'll try. And then this is a snap top. So I'm going to grab a hold of it, and I'm just going to snap, and that breaks completely off. And now this turns into a dropper bottle for me. And I'm going to zoom in the next video, and I'm going to apply the dropper bottle at the circle area of the cartridge, because that's where the sample needs to go. All right, so here's my HEMA trace card, because guess what? It picks up traces amount of blood. That was creative, wasn't it? All right, so I've zoomed in a little bit on the cartridge. Again, my sample is going to be applied in the well at the bottom, and you can see the T for test and the C for control. So these directions are going to say, all right, we are going to apply 3 or 4 drops of that buffer into the well, and I just did that. So by no means do you need to use all of that sample in that little dropper bottle. 3 or 4 drops is all that you need. And then the directions are going to say, all right, now when you do that, after you've applied your sample to it, 3 or 4 drops is all that needed. Sometimes you have to wait for 10 minutes. And then after 10 minutes, you come back and you take a look at what the card is actually doing. So with that said, what will happen is that you're already seeing some changes. But what is going to happen, I'm going to stop the video here, and I'm going to await the appropriate time, read the results 10 minutes after the sample was added, and we will come back and we will take a look at what this card looks like. So the magic of lab videos, I'm going to fast forward, and then we're going to look at 10 minutes into the future. All right, everyone, we've waited 10 minutes, and as you can tell from our pregnancy test, just kidding, our blood test, our cartridge shows 2 lines. So once again, like I had mentioned in a previous video, this very top line that's beside of C is control. So that means the cartridge is working the proper way. So we have to have some type of quality in place to make sure that that's going to go forward, and that is the C for control. So the control band is present, and then down here at the very bottom, that line is beside of the mark that says T, and that is our test sample. So I see 2 lines. Control is working, and the test sample was positive for what I just swabbed. So that means that according to the HemaTrace card, that is a blood sample. All right. So again, not quite presumptive. It's very much more specific. It plays off of antibodies and those types of things. So the reagents are very general. A lot of things can throw them off. A lot of things can give you false positives. But with the HemaTrace test, with the cartridge, all of that really goes out the window. It really kind of is blood now. So what we want you to do is find a blood sample first and use Kastle-Meyer first. When you find a positive blood sample with Kastle-Meyer, use that same blood sample and keep testing it over and over and over and over and over, and then end up with 1 HemaTrace and 1 HemaSticks test for that particular garment. So you're actually going to have probably more than 1 garment that you're going to have to do that with, but keep in mind all of these just require 1 and you're done. All right. So on the data sheet, you are probably going to see schematics that look like this. You know, there's one that looks like a pillowcase maybe. That's the one that I just did. But you're going to have a drawing already on a sheet of paper, and it's going to want you to circle the area that you found the blood sample in. So front or back, right? So you're going to see a front and a back. We want you to just draw a random, not a random, but a circle in the general location of where you found that blood sample. Don't think that you got to be too exact, just the general area of which you found it. And then down here at the very bottom, you're going to see, all right, well, what did it do with Kastle-Meyer? What did it do with LMG? And what did it do with TMB? And it's going to ask for color and then results. Does that mean it's positive or does that mean it's negative? It's not on the table right now, but you'll also see 2 more lines. An additional line will be for your HemaSticks, and an additional line will be for your HemaTrace card. I'm also going to probably ask you to take pictures with your cell phone, so that way we've got proof that you did the test, and we've got proof that your color changed the way it's supposed to, so that way you just don't randomly write down things in your data table down below. So keep that in mind as you do the test. Don't throw things away. You'll need to take some pictures and upload them into Canvas. All right, guys, and in my final video, I just kind of want to show you maybe more what will be in your kit, basically, and give you more tips and tricks. You just saw me do something with a white pillowcase because I thought it showed up a little bit better, and you could see the different colors of reds and browns. What you'll find, though, are colored garments most of the time on the inside of your lab packets. Once more, keep in mind that this is a pair of male underwear. Very big ones, I might add. And then on this underwear, you will see a lot of bright red. So bright red is here, and bright red is going to be there. That's probably not blood. It could be something else, but it's not blood because, again, older blood or dried blood that's been exposed to the air for a little bit will not be bright red like that. But there are some areas on here that are very suspicious looking to me, and some of these are more of the dingy spots that I see here. This one's not as dingy. That's a little bit deeper. It's a little bit darker. So that one possibly I would test. These faint ones, those are possibilities that that's blood as well. If I come down here, kind of in the groin area, I'll see a spot here and a spot there. So those are going to be the areas that I'm going to swab first if I had to do this. But keep in mind, if I get my Q-tip wet and then I rub the spot and it is bright red in the very beginning, then that probably means that's not really where I'm looking for or what I'm after. If I flip this on the very wide side on the rear end, I'm going to find hardly any spots at all back here, which is okay. There's nothing back here for me to kind of be suspicious of. I always kind of want to look at the front side and the back side of these articles as I'm testing them, because these blood splatters can be located anywhere. And also keep in mind that they are not in the same location for every single garment that we put in your lab kit, meaning that your garments could be unique to someone else's garments. So that way they're not in the same location and one person finds it and shares it with everybody else and everybody else has an easy A and go, wee, we get to enjoy our weekend. That's not how this is going to work. I don't want you to enjoy your weekend. I want you to be miserable and sufferable. No, just kidding. All right, so with that said, good luck on the presumptive test. It's just some general reagents that we've got to worry about. Keep your lab gloves on, keep your lab diaper out, and make sure that you keep everything clean. You're more than welcome to do this in our lab. If you want to do it in our lab, we have these kits ready to go. Or this is kind of maybe an online class, and if you can't stay around and do it in the lab, take it home. Just be okay with getting your countertops all nasty and trashy. So with that said, good luck. If you need any questions answered as you do the test or before you do the test, please let me know. But I think that with the help of this video and some of the lecture notes, I think that you will be perfectly fine in this lab activity. So good luck. Make me proud. Get an A.